Assalamualaikum and hi. Today our group will present about monkey cloning. How was monkey cloning started? It starts when a team of American scientists has produced the first non-human primate clone.
This is Tetra. Tetra is a rhesus macaque baby. She was born in September 1999 at the Oregon Regional Primate Research Center in the US.
However, the creation of Tetra was not done by nuclear transfer as the leadership was. Instead, embryo splitting was used to create identical embryos, in which Tetra was one. She is the first cloned primate by artificial twinning. Embryo splitting involves dividing an egg cell embryo into 4 identical parts. It has been done before in mice and cows, but this is the first time a monkey, the closest genetic relative to humans, has been cloned. Scientists believe identical monkey will be the best model for studying human disease. It is completely different from the cloning of the leadership, but both experiments have something in common, that is their contribution to growing debate about the ethics of cloning.
Tetra was created using embryo splitting, a process where 2 cells in the embryos are split at the 8-cell stage to create 4 identical 2-cell embryos. This was the first time that this technique had proven successful in monkeys. Although it is often used in cattle, she was the first primate to have been cloned using this splitting technique. The first non-human primate derived from nuclear transfer was created in 1997 using a different technique for cloning. Only 2 of the 4 embryos survived to a sufficient stage in which they could be implanted into surrogates, and Tetra was the only one to be delivered successfully after 157 days. The announcement of Tetra was made on 13 January 2000. When she was 4 months old, it was thought that by producing identical primates, advances in human medical research could be made. A further 4 monkeys cloned using the same technique were due to be born in May 2000.
The aim of the research and development of these true clones is to produce models for testing whether it is possible to grow stem cells for transplant into patients with failing organs. Most new medical techniques are tested on mice. Monkeys, being genetically more similar to humans, may provide more reliable results. With genetically identical monkeys, researchers could be more confident that any effects they see are caused by the test animal's environment or are the results of the experimental therapy. However, the research has inevitably sparked the debate over the possibilities and implications of human cloning and the ethics of human stem cell research. The scientist named Sternberg said the application of embryo splitting to monkeys could help researchers cut the number of animals they need for their work. For example, he said, AIDS researchers know there is a tremendous variety on the response of monkey to HIV, the virus that causes acquired immune deficiency syndrome.
I would like to talk about pros and cons of Tetra, which is embryo splitting. The pros. First, it can grow stem cells for transplant into patients with failing organs. Second, it is valuable to medical research as it may reduce the number needed for some experiments. Third, it creates new opportunities to study disease and new treatments. For the cons, there are 2. First, causing some damage that prevents the embryos from implanting and developing properly in the surrogate's mother's uteri. Second, which is the last one, a limited number of identical offspring.